Prophase is the first step of the mitotic phase, and it usually takes the longest out of all phases. First, the chromatin condenses in the chromosomes, then the centrioles move to opposite ends and start making spindle fibers. While that happens, the nuclear membrane is breaking down, and the nucleus disappears. Metaphase is the second step of the meiotic phase. The spindle fibers that were made in the prophase step are a protein structure that helps divide genetic material in the cell equally. They attach to the centromere on opposite sides of the poles, and the chromosomes move to the equator as the nucleus membrane is completely broken down. Anaphase is the third step of the meiotic phase. In this phase, chromosomes are split and move to opposite ends of the chromatids, and they also move to the opposite ends of the cell. Telophase is the fourth step and technically the last step of the meiotic phase. In this phase, the chromosomes go to opposite ends of each of the new formed cells, and a nuclear membrane forms around each new cell. The cleavage furrow is caused by the contractile ring, which is at the equal cortex and goes inwards to separate the two cells. Cytokinesis is the process that divides the cytoplasm from the parent cells into the daughter cells. In mitosis, one parent cell splits into two daughter cells. The two cells are identical to each other and the parent because they each contain the same amount and type of genetic material. The nucleoli reappears, and this step is different between plants and animals because an animal forms a cleavage furrow, but because of the hard cell walls, plants can't do that. The cancer drug that I research is Pradaxel. It impacts the G1 phase, which is in the interphase. It stops the microtubule structures, the structures that help divide and replace the cell, from dividing and replacing the cell. If the cells cannot divide or replace, it dies.